Okay. Week 6. So we're going to be doing chapter 16, which is where I'm going to spend the most time, quite obviously. And then chapter 17 and 18. Chapter 17 deals with our disorders of the immune system. We can't really understand that and what the implications are with that without understanding our immune system. So that's why chapter 16 is going to be so key. And then chapter 18, I feel like it's pretty straightforward. It's not too difficult for most people, but it's our diagnostic testing. And it's more about categorizing our diagnostic tests. But to start with, chapter 16 dealt with our adaptive immune response, also known as our acquired immune response or specific immune response, basically our ability to respond to two specific antigens and eliminate a specific threat, but also have memory as a result of this. And this is the key of this whole thing, is the fact that we will have memory. So it's the go-to of our third line of defense, which is our adaptive immune response. So to begin with, all of our lymphocytes start in the bone marrow. They all start there. And the B cells will continue to mature there, whereas the T cells will move to the thymus and then mature there. And once they have fully matured, they'll then be naive B cells or naive T cells. And then they will be able to move. The naive B cells will move into usually the lymph nodes and kind of camp out there and screen things as they come through the lymphatic system. Until they get the right signals from their T helper captain to tell them they can make antibody. Whereas the T helper cells and the CD8 T cells, they kind of move throughout the periphery and go throughout the body, just trying to see if they can find that specific bracelet or that specific antigen that they will react to. Once the lymphocytes are activated, whether you're a B cell or a T cell, the first thing you do is proliferate. You multiply, you make a ton of copies. So basically you're a T cell that is naive, T helper that is naive. And the first thing you do is make a whole bunch of T helpers that are naive, just general T helper naive cells. So just basically T helper, T helper, T helper, T helper. And then after you've made a whole bunch of these, some of those, we'll make 4 of them. Some of those will turn into memory. I hate that. I hate this. Just zoom out. Thank you. Some of them will turn into memory, some of them regulator, and some of them effector. Always, it's those 3. So some of those that we proliferated out will then differentiate into memory. Some of those that we proliferated out will turn into regulator, and then others will be effector. So I would put E here, but by that, I'm not really referring to effector, right? Our effector cells for our T helper cells are more specific than that. They are going to be our TH1 and our TH2. These are our effector. The ones that are actually doing the job of that particular cell type. Okay. So we proliferate first, and then those proliferated cells will differentiate into that. Just to clarify that. No. So. Okay, so clonal selection, once you have made your repertoire of naive lymphocytes, whatever they are, once you've made that repertoire, they will go through something called clonal selection, which is our process of removing anti-self. And then the whole process of only proliferating out the one that sees that specific bracelet, not any other bracelet that was similar to it, but that specific bracelet, or really honestly, if we're being scientific about it, that specific antigen. We're only proliferating out that one. So if you go in to a body and you activate a T cell that sees the flu spike protein, you're not going to activate all T cells, right? You're just going to activate the T cell and proliferate out the T cell that only sees that flu spike protein, not ones that see COVID spike protein, not ones that see HIV spike protein, but specifically that antigen. So that's the idea of clonal selection. We've removed anti-self, and that's the clonal deletion process of clonal selection, removing the anti-self ones early on during development, and then only proliferating out the one that's appropriate for that antigen. That all together is clonal selection. All right, an antigen is any specific molecule or thing that your body could see as foreign. And it can be very, very specific down to a part of a pathogen, or as well as very more general things like even LPS could potentially be seen as an antigen. And we would only see it as an antigen or an immunogen whenever we are reacting to it with our the third line of defense, our adaptive immune response. But if you're capable of provoking the third line of defense, your adaptive immunity, then you are an antigen or an immunogen. Okay. So even though your flu spike protein, if we look really close at the surface of the flu virus, and your spike protein might look like this. Maybe an antibody, and each one, I don't know, one has a circle, one has a square, circle, square, who knows? But your antibody might only see as an antibody. We know antibodies look kind of like a Y, and they've got a couple of lines coming, right? Your antibody might only interact with part of that spike protein. The part that it actually interacts with is called the epitope. The part that your immune system is actually interacting with is called the epitope. So the antigen might be the flu spike protein, but the part of the protein that we're actually interacting with is called the epitope. Some epitopes are too small for us to trigger an immune response, and so those are called haptens, and they will need a carrier molecule in order to help our body react to it. So let's say that you are this tiny, and our antibody, it just can't interact with it. I'm drawing it really janky, but whatever. Our antibody can't interact with it, but then we stick it onto something else, and now suddenly our antibody has something it can help grip onto with that. And so this is called the carrier. And so this is something that we use as a mechanism in creating our vaccines. When we have something like the tetanus toxoid, I wish this was more intuitive. When we have something like the tetanus toxoid, which is a toxin produced by Clostridium tetani, it is too small on its own to elicit an immune response. But we pair it with other things to act as carriers. And so those carriers are things like, it happens to be other antigens, and they all work together to help us react against the tetanus toxoid. The Tdap vaccine, tetanus toxin, diphtheria toxin, and pertussis toxin. They're all, most of them too small for our immune system to interact with on their own. But when we put them all together, suddenly they're big enough that we can actually get antibodies to each of those. And they all work together as carriers for one another for our immune system to help activate itself. So that's how the haptens will work. They're too small by themselves. Once we have antibodies to them to help activate the response, none of the antibodies exist to act against that too small thing. It'll be okay once we have the antibodies activated, but that activation response, it just needs more for us to be able to present it on the MHCs and the things like that. So that's the whole point of the carrier molecules for that. An alloantigen is anything that is normal in self to one person, but would be seen as foreign to another person. My MHC looks like self to me, but your MHC probably looks like foreign to me. My body can still recognize it as an MHC1 molecule, but it would see it as foreign MHC1. So it's to my body, your MHC1 that's just chilling and not doing anything. I'm not infected inside. I'm just chilling. My body would see that as MHC1, but something's going wrong with you. You must be presenting something foreign. And so my body would attack that. And it's getting the signals telling it that it can go ahead and do that. So those are alloantigens. MHC1 is a very common one. It differs from person to person. We actually call MHC HLA or human lymphocytic antigen. And it is one of those markers that we use in basically determining for organ transplant, which we talk about in chapter 17 a little bit, but MHC is very important for that. Then there are super antigens. These are antigens that elicit a super strong response that is out of what we would expect to be necessary. So when we look at endotoxin, LPS is a pretty common one for this. It causes a massive, massive cytokine storm in response to it being in our body. It's way overreacting to that thing. And in fact, COVID, spike proteins and other aspects of COVID virus are pretty similar. They can cause our body to overreact and create too much of an inflammatory response and too much of an immune response. So these are all super antigens, things that cause overreaction by the immune system. And some of these things are what are responsible for like toxic shock syndrome, like we learned about with tampons. And those molecules will basically trigger our immune system to overreact. Not appropriate. It's not appropriate for what is being introduced into the body. It's too much. Antigen presenting cells. Our antigen presenting cells are our monocytes and their derivatives. Remember, monocytes are in your bloodstream. Once your monocytes move into a tissue, they differentiate into either macrophages or dendritic cells, depending on the tissue. So monocytes and their derivatives, macrophages and dendritic cells, as well as B cells. So B cells basically take up house in your lymph nodes and filter out your lymph. And they sit at the checkpoints, which are your lymph nodes, basically checking everybody's papers, making sure that they're good to go. Once they see somebody's paper that they match with, hey, your paper says that you are foreign and you are, you are specifically Jason Voorhees or something like that. You're Jason Voorhees. I was made specifically to attack Jason Voorhees. So get ready, we're getting ready to start a fight. So B cells are waiting to see their specific thing. They have on B cells, this is a B cell, receptors that look like this. This is a B cell receptor. A B cell receptor looks strikingly, as I'm sure you can imagine by my wonderful drawing here, looks strikingly like an antibody. And in fact, it is. It's what we call an immunoglobulin. As a receptor that is bound to the surface, this immunoglobulin is IgD. That is what IgD is. It is the B cell receptor. And what happens is this B cell receptor happens to see its foreign thing, that it reacts to Jason Voorhees. It sees, oh, I was made for Jason Voorhees. I'm reacting with Jason Voorhees. And now I'm reacting with that. It can't make antibody and secrete this in its secreted form, which is first secreted form is IgM, which is a pentamer, which I'm going to try to draw it, but bear with me, okay? So the first immunoglobulin that it secretes that isn't bound to the surface is a pentamer. It looks like this. And this is IgM. But it can't do that until it gets a signal from the Th2 cell, the captain. Okay, well, it's not my day. Needs the Th2 signal. And now I'm zoomed out, and yes, it won't let me erase unless I'm like properly zoomed. Promise this isn't going to be frustrating. Needs Th2 to send its signal to activate it so it can make the IgM. It can't do that without the captain. Okay, but while it's doing that and collecting the antigen through the lymph node, it's acting as an antigen presenting cell. And so what it'll do is, I've seen Jason Voorhees. I picked this up outside. And so it'll take in its little IgD and put that piece of Jason Voorhees on MHC2, which it found outside. And now it can interact with the naive T cell and activate it and get the signaling going all simultaneously. So it can act as an antigen presenting cell that way, while also getting in that interaction its own stimulation to start making antibody and everything. So that would lead to the activation of the proper captain and the activation of the B cell and everything that would be happening downstream after that. So waiting for Jason Voorhees there. I don't know why I went with Jason Voorhees. Isn't Jason Voorhees like Friday the 13th or something? Something like that, I think. Anyways, that's just the name that came in my head. I guess that's the kind of mood I'm in. Whatever. So MHC2 presents exogenous antigen always. So this is the outside stuff. And MHC1 presents stuff that was endogenous. This is the inside. So there are two types of MHCs. Any cell in the body can get infected inside with a virus, right? And so every cell in the body needs to be able to call out for help, that I'm having something going wrong inside. But only our antigen presenting cells, which we introduced previously as being our macrophages, our monocytes, our dendritic cells, and our B cells, only those guys are going around eating up antigen and saying, I found this outside. Not every cell can do that. So our antigen presenting cells, they are the ones that have MHC2. They're the only ones that have MHC2. But any cell needs to be able to cry out and say, I'm infected inside. And then we can target those cells that are infected inside and kill them with our CD8 T cells if we have the captain that allows them to do that, that was already acted with the outside stuff. So that's where that whole process that I talked about prior to the lab came into play, right? So we'll kind of go through that a little bit again in a minute. But this is just get our concepts out of the way first. All right, so our T helper cells, we have two types of T cells, T helper and CD8 cytotoxic T cells. Those are our main types. Yes, we have memory, and yes, we have regulatory. I'm way less concerned with that, okay? But the regulatory ones, obviously what the regulatory ones do, whatever kind of lymphocyte you are, whether you're a regulatory B cell or T cell of any kind, the regulatory guys basically control inflammation. That's their whole point. The regulatory guys keep things from getting out of control. Okay. So Th1, they are still CD4 positive. They still had to get activated by outside stuff. So always CD4 always sees MHC2. I'm just write a 2 so that it's not going to be. And CD8, it only and always sees MHC1. That's the whole purpose of those molecules. CD4 interacts with MHC2 and CD8 interacts with MHC1. That's the whole point of those CD things. It's a purpose. So if you are a CD4 Th1, you have CD4. You never have both. So if you're a CD4 Th1, you see MHC2. But you're going to send signals to your CD8 cells. The CD8 sees the MHC1 for the inside stuff, but it can't do anything until it gets the signal from the Th1 cell. All right. Th2 recognizes, this is the part that made everybody confused. So we're just going to like block that out. Focus on the details here. Because yes, MHC1 plus antigen doesn't interact with the CD4. MHC1 plus antigen activates the CD8 response, which is modulated by the Th1 cells. So that's the part that I think a lot of people are getting confused on. So yes, CD8 sees MHC1, MHC1 plus antigen. Wrong. It's this kind of response that we're interacting with. MHC1 plus antigen activates CD8 cells only if Th1 cells send the signal, give the go-ahead. That's really what that is talking about there. So just to be clear, that's really what that whole section is talking about there. I wrote it in like the worst way to confuse everybody as much as I could, apparently. But yes, we're clearing that up now, okay? CD8 sees MHC1, and the CD8 cell can only kill the MHC1 presenting thing, MHC1 plus antigen. It can only kill that cell if Th1 cells give it the go-ahead. And the Th1 cells are CD4 positive, period. Again, we've gone over it once, but we'll go over it again after we go through our details here. B cells can make memory cells and regulator cells, and their effector cells that make antibody are called plasma cells. Plasma cells are B cells that produce antibody. Antibody looks like this. 2 long pieces, which we call the heavy chains, and 2 short pieces, which we call the light chains. The light chains and the heavy chains, basically like the part that interacts with the antigen. Let me get another color here going. I don't like that. Okay. This part here interacts with the antigen. Right here at the very tips, right? That's the part that interacts with the antigens. That's where the most variability is going to be. This whole part up here that I have circled in the big orange circle, this is called the FAB, the FAB fragment, also known as the variable region. These are the interchangeable. All right. This part down here in green is known as the constant region or the FC. We know that the variable region interacts with the antigen. That's its whole purpose, is to see the specific antigen. But the FC, or constant, I like FC, is C and constant. So we have A, B, and C. A, B to me is antibody. What's the whole point of the antibody? To see the specific antigen. So that's the variable region. But the FC is the constant region. What's the point of the constant region? The constant region determines the function of the antibody. And so when we talk about the different types of antibodies, IgG, IgM, IgA are our biggies. When we talk about IgE in the next chapter the most, it's more associated with allergies and parasites. And IgD is our receptor that we introduced up here, the B cell receptor one. So the whole way that we're dictating it as IgG, IgM, IgA, IgE, or IgD, and what functions those play in the cell and in the immune response is determined entirely by that FC region. Your IgM because you have the IgM FC region. Your IgD because you have the IgD FC region. And so the FC region, the constant region of IgD, means that it's going to be a surface receptor. IgM FC region means you're going to be the pentamer and you're the first one that we make. You know, if you have the IgG FC region, that constant region determines that you're the workhorse. You're the one that's going to be present in the bloodstream, circulating throughout the whole body, providing a lot more functionality than IgM. But we always make IgM first. It's just what we make first. It's what we have readily available. And then your B cells will go through class switching. So you always make, obviously you have IgD as your receptor first, right? But that's not secreted. So the first one that you're going to secrete out into the bloodstream is IgM. And then IgM first, and it is limited in its ability to function, but we can make it right away. Then we'll go through class switching. While we're making some IgM and trying to get things underway, we'll class switch to IgG, and we'll make new memory cells that can just make IgG. And so that the next time that we see the thing, we'll make IgG right away. And it's so effective and so efficient. It'll make up 80 to 85% of your, all of your antibodies will be IgG. And they can do all of the things that antibody can do, which includes the things above: neutralization, opsonization, coating, agglutination, and complement activation, and acting as antitoxin. So I'm going to walk through each of those functions. See if I have some space. We'll do it up here. We'll do it off to the side over here by these guys. So antibody functions. Antibody function. I don't like this color. It's too light for me. We'll do blue instead. Antibody function. Hard for me to see it. All right, so our antibody functions. Neutralization is basically like you're a virus and you've got your spike proteins that help you dock with your whatever. You have a circle shaped spike protein, and then there's some sort of molecule on a cell, target cell. And normally you would dock like this, but the antibody comes in and blocks that. That's neutralization. All right, cool. Next, I can't erase if I'm zoomed in and I don't want to zoom out. Okay, next, opsonization. We've introduced opsonization before. It's basically like, opsonization is basically like you act as an opson, which is what our antibodies do. Here's your bacteria cell, and it's got that big old capsule around it that makes it hard for our phagocytes to eat it up. It can't grab onto it. So this is our macrophage. And this is our bacteria with a capsule around it. It can't eat it up very well. But our antibody comes and binds on, and now it acts as a handle to help the macrophage grab onto it. Molecular handle. So the antibody sticks to the capsule. The macrophage can bind just fine to that antibody. It couldn't grab the capsule by itself, but it can grab the antibody just fine. So now we have something to grab onto to help us eat up the bad guy. So antibody can do that directly. Antibody can also activate complement, which to remind you guys from chapter 15, what complement does. Complement either activates the inflammatory pathways, or it creates that membrane attack complex, which pokes holes in the target cells, causes them to lyse and leak out and burst open, or complement itself can act as an opson, helps with opsonization. It's a molecular handle. So antibody can activate complement, and complement, if the antibody's not doing a good enough job as a molecular handle, maybe the complement will do a better job. But you'll also activate complement to poke holes in the target thing. If antibody's bound on there, complement's like, hey, we need to poke holes in that. And it'll also be like, complement will also say, hey, there's antibody there. We'll also activate the inflammatory response. So complement's helping aid in all of those things too. So we have done Neutralization, we have done opsonization, and we've done complement here. All right, coding. Basically, you're a big old nasty bacteria and you're trying to get stuck to tissues or establish your infection place or whatever. Antibody just coats these guys. And now you can't, you're having a hard time sticking to things. I'm drawing my antibody weird, but you get it. Whatever. It just covers it up. And so now it can't stick to surfaces. It can't get into deeper tissues. It's having a hard time functioning or interacting with other bacteria even. Coating is just covering it so it can't get around very easily. I've seen like these videos in some movies, like sci-fi movies, where like they'll have a security system that'll just stick big sticky balls to people, and then the people are like stuck to the ground and they can't move. It's sort of like that. It's basically like covering up these bacteria so they can't get around, it can't interact with their environment well, which is a little different than neutralization, which is more specific blocking of a specific interaction. General coating and covering versus neutralization, which is specific blocking of an interaction. Antitoxin, I'll go ahead and bring that one up next because antitoxin works in a similar way as neutralization. Whereas neutralization, we have antibody blocking a specific part of a pathogen from doing its job, and coating just covers it generally. Whereas antitoxin, we are binding to the toxin that the pathogen makes. And so that would be, for an example, like tetanus toxoid. When we have antibody that blocks tetanus toxoid, it blocks the tetanus toxoid from doing its job in our body. It blocks it from having its functionality to cause us to have spastic paralysis. So we can actually block that from having its function there. So we're not blocking the pathogen or a part of the pathogen, we're blocking a protein that the pathogen secreted. So that's antitoxin. And then agglutination. Basically what we're doing is we can have a bunch of bacteria that are hanging around near each other, and then the antibodies come in and then cause them to stick together. So they're binding at multiple sites on these bacteria, causing the bacteria or the viruses to stick together. So when we have cells that are doing that, we call that agglutination. Precipitation is whenever we have proteins or smaller molecules that we do that too. All right. Antibody titer is a measure of the concentration of antibodies in serum. The higher the concentration of antibodies, the more recently that person probably saw that antigen. So the higher your HIV antibodies, the more recently you have seen active HIV in the bloodstream. The lower those antibodies, the less recently that you've seen active HIV in that bloodstream. The primary response is whenever we start, we got to remember, right? You don't just start making antibody right away the first time that you've seen something. When you very first see a virus get into the body, what did we have to do in our example? We had to have the macrophage eat it up first, right? So first things first, the pathogen gets into the body. Whatever cells are there, usually your mast cells or cells that are getting infected, are calling out for help from our innate immune system. And so the innate immune system attacks first. This is usually your neutrophils. And as the neutrophils are eating things up and recognizing PAMPs, they're calling in for signals from macrophages and stuff like that. Then macrophages are our first antigen-presenting cells to that site. So after we've called in the neutrophils, after we've called in the macrophages, now the macrophages can pick up the antigen and go and present it to our captains and start activating our captains. The captains have to be activated first, otherwise we don't get antibody, we don't get cytotoxic response, right? So we will send out our macrophages, they pick up the bad thing, they find the right captain, the right T helper cell. They activate the T helper cell. Well, that T helper cell now has to proliferate and then differentiate before it can activate making antibody or cytotoxic T cell response, whatever needs to happen next. And that crap takes time. We're talking about going like a week or so until you get your proper lymphocytes multiplied out and ready to fight. And remember, your first antibody that you're going to make is going to be IgM, which is less effective. So IgM, we're going to make that first, but we're trying to work really hard to get to IgG. So that's going to be another few days until we get IgG. And once you get IgG, you'll clear out whatever infection you have easy. But that's typically going to take a week to a week and a half to get to IgG. So that primary response has a latent period where we're activating our response, where we're activating our T cells and getting all of them multiplied and proliferated and everything else. And we'll start with IgM and eventually switch to IgG. Whereas our secondary, also known as our amnesiatic response, our memory response, our memory cells that made IgG They already know how to make IgG. So we can make that right away. So it's a faster response. We can make it immediately. We don't have to go through the proliferation and differentiation bullshit. We can just go straight into making IgG at a much higher level with a much faster response. All right. This is why vaccines work the way they do, because we're relying on that memory to just start cranking out IgG as opposed to waiting for your body to start reacting to a thing. Not to mention when we talk about having measles, when you have the measles virus, measles has some aspects of the disease that basically inhibit your body from creating memory. So you can have measles many, many, many times and never have memory. But we've designed the vaccine to help promote making memory. And so that's why the measles vaccine is so effective at preventing the spread of measles. Anyways, moving on to the different types of immunity you can have. These are pretty straightforward, so I won't spend a lot of time on them. Natural, active. You made your immune response to something that you saw in nature naturally, and you had an antibody response of your own. So natural, you saw it to usually exposed to an illness, whereas artificial, you saw it introduced because of medicine. So this is like vaccines. Active, you made your own immune response. You made antibodies. So I had the chickenpox when I was little. And now when I go around people with chickenpox, I don't get it again, right? Because I have natural active immunity. I had it naturally, and I have active antibodies against the chickenpox because I've had it before. Same thing with mono. I had mono. I developed antibodies to it. I can't get mono from other people now because I have antibodies to mono. So that's natural active immunity. Natural passive, taking the antibodies from somebody that made them naturally and naturally giving them to somebody else. So the best example we have here is mother to child during breastfeeding. In the colostrum, in the breast milk, you have mother's maternal antibodies that she made naturally to something that are getting passed to the baby naturally through nature. But the baby isn't making the antibodies, right? It's being supplied to the baby. So that's a passive immunity. Whereas artificial passive, medicine is introducing antibodies that we collected through medicine from somebody else. Now, they may have seen the disease naturally, or they may have seen the disease from medicine. That part doesn't matter. But the fact that we are taking antibodies that we collected from medicine and medically introducing it into somebody, that's artificial passive. So this is gamma globulin infusions. Even things like Humira and other monoclonal antibody treatments, where we're giving antibodies to somebody to protect against something, those would also be artificial passive. All right, and then we talked about the different types of vaccines. You can have live attenuated. This is a live virus or live bacteria that can't multiply in the body, but it isn't technically inactivated. It's just not supposed to cause disease. So it can live, it can multiply potentially, or it might be somehow deficient in causing the disease somehow. So that's attenuated. It's a live thing. It can multiply, but it's not supposed to cause disease in people. Can they revert back? Yes. So that's a problem with polio vaccination that we sometimes see with the nasal vaccines that we use in third world countries to vaccinate against polio, that it can sometimes revert back to the original disease-causing form. We don't use a ton of live attenuated stuff with bacteria. We mostly see this with viruses. Killed, this is where the pathogen can't multiply or anything. We're just using it as a source of antigen. mRNA, like we have with the COVID vaccine, we're introducing mRNA. The cells translate the mRNA. They put the spike protein on your own arm cells, and then your body sees that as foreign, and it creates a memory response against it. This has been useful in COVID, not to prevent infection, but to train our bodies not to overreact to the infection. And so now when you get COVID and you've been vaccinated, your body reacts to it like it's the flu or like a cold, instead of being overstimulated, causing cytokine storm, which is what was putting otherwise healthy people in the hospitals with COVID. Subunit, using just part of a pathogen instead of the whole pathogen or something that codes for that part. Toxoid, if the toxin is what is causing the disease, then we can create an antibody to the toxin. And that's what Tdap vaccines do for tetanus, pertussis, and diphtheria toxins. Conjugated is where we have to link it to something else to get a better reaction to it. That is kind of also what we see with our toxoid vaccines, our Tdap vaccines, but we also have other vaccines that are going to rely on conjugation that are just by themselves. We can introduce vaccines either intramuscularly, subcutaneously, or intradermally, or we can have oral or nasal vaccines. Intramuscular is really literally what it sounds like. You want to inject the vaccine into the muscle tissue. Some things work better subcutaneously, where we will just want to do it just into the like dermal layer, but not into the muscle quite. Whereas intradermal is where you'll usually scratch it. So usually like smallpox, which we don't do here in the United States unless you're in military, is where we'll create scarification on the arm. And then that scar process is what causes us to have the proper immune response with that. So we don't see a whole lot of intradermal vaccination, but they do exist. And then obviously oral and nasal, we're going to be squirting into the nose or into the mouth. We see this a lot with like Bordetella vaccinations with kennel cough with animals and stuff like that. And people who are allergic to the flu vaccine, we will give them a version that's an oral nasal version. It's usually live attenuated. All right, adjuvants are things that enhance the immune response. They're chosen for a reason to do the thing. One thing that I've been hearing a lot about with the COVID vaccine is that it has propylene glycol in it. It does. Propylene glycol, just to be clear, is antifreeze. Okay, so it is a toxic substance, but it's important because it helps the cells take it into the cells. So if I give you too much propylene glycol, then that can be toxic. Of course it can. So some of these adjuvants can have negative side effects, but they're more likely to have a positive effect with the vaccine. And so it's the risk versus benefit thing that we will have to outweigh with that, which is the case with a lot of our side effects with our vaccines. And being aware of misinformation about that stuff is important as well. Anytime that we are vaccinating somebody, it's your same immune system that would be reacting to the natural thing. We're just using our knowledge of the immune system and how it behaves in order to properly control the response and control the outcome there. Herd immunity is the idea of a certain number of people have to be vaccinated in order to protect the population that isn't or can't be vaccinated. With the measles, for example, which is the big one, measles is the most contagious disease that we know in mankind. And 97% of the population needs to be effectively vaccinated in order to protect the other 3%. And we're just not getting those numbers, which is why we're seeing an increase in the spread of that. So herd immunity is protecting those populations that can't get vaccinated by having other people vaccinated. All right, moving on. We'll come back to I'll walk through the immune response once we get through our concepts for chapter 17 and 18. We'll do just one big walkthrough and make sure we got that all cleared out. But Chapter 17 concepts. First, we have the hygiene hypothesis. This is the idea that in a lot of our first world or developed countries, we are not exposed to that many antigens and foreign things and whatever. And so some aspects of our immune system weren't trained properly. And so we're more susceptible to allergies and things like that as a result of that. There's a lot of evidence that supports this. For example, in underdeveloped countries or third world countries, where they still get parasitic infections, they don't have allergies, which is wild. But yes, this is true. So when we think back to like the history of humanity, it's like, well, why didn't our ancestors have, food allergies or whatever? Well, they had worms. That's why. So the worms were training their systems to react appropriately to those things. So we don't see a lot of documentation of food allergies or any allergies at all, really, from older populations before hygiene was properly practiced, because we didn't know that our immune systems were being trained properly to handle that stuff. So that's the hygiene hypothesis, where we are so clean that our immune system isn't getting trained properly, and so we end up with allergies. So we have 4 different types of hypersensitivities and 2 types of hyposensitivities. The hyposensitivity is also known as immunodeficiencies. So I'll write that down there. So there's 4 hyper and 2 deficiencies. So our hyper are overreaction of our immune system. The first one is type 1. Type 1 is governed by IgE. So remember that this is that antibody that's usually associated with parasitic infections. But in more hygienic countries where we practice a lot of hygiene and are more aware of medicine, IgE is more commonly associated with our type 1 hypersensitivities. And so this is going to be either atopic, which is local, or anaphylactic, which is systemic. Atopic is local and anaphylactic is systemic. So some examples of atopic allergies would be asthma, hay fever, and eczema. Eczema is known as atopic dermatitis. And so you have actual allergic reaction within the dermatitis, within the derma, which is the skin, governed by IgE. Whereas anaphylaxis is you're going to have it through the bloodstream, not just the skin. You might have, eczema all over your entire skin, over your body, but it's still not systemic. It's not affecting your internal organs. It's not affecting your blood. It's not affecting all this. But anaphylactic is systemic, so it's going to be throughout the entire body. It's just going to have much more quicker and much more severe reaction there. The different types of allergens, inhalants, contactants, injectants, and ingestants, you can have also crossover into groups. It just depends on how that person was exposed at that time to trigger their reaction. And that can also govern whether it's going to be atopic or anaphylactic as well. I'm not going to go through any of that. That's, I feel like, pretty self-explanatory. Because this relies on IgE, this involves an initial sensitization. Where we were talking about our primary and our secondary response with our antibodies, you had to see the thing and see it as foreign and go through proliferation and differentiation with the B cells and the T cells and all of that in order to get IgE at the end. So your very first time that you ever saw a bee sting, you might not have had a reaction. So most people can get stung by a bee once and not have any reaction whatsoever. But whenever you get stung the second time, now your body's ready to release the histamine and everything associated with that because you can make IgE right away in a large amount. And so what happens is we get these mast cells, which we've talked about previously. These are one of our granulocytes. And they are just camped out in many of our tissues all the time. And your mast cells, they wear IgE like a little flag, like a receptor. And so this might be IgE that sees pet dander. And then pet dander comes along and binds with it, and it causes the mast cell to release things like histamine and other inflammatory. That's what mast cells do. And then that's where your type 1 reactions are going to come from. So it's the mast cells that are causing the problem, not really the IgE itself. And so the mast cells that are carrying around the IgE as a little flag, as a little, receptor, causes the degranulation of those mast cells. And this will release histamine and, other things like bradykinin and all sorts of stuff that are involved in inflammation and airway constriction and whatever. So, we can use antihistamines to treat what we usually use antihistamines to treat the atopic. We can also use corticosteroids to treat atopic. More commonly, we're going to use epinephrine and adrenaline specifically for systemic, also known as anaphylactic reactions. We're not going to usually reach for adrenaline unless we are treating the systemic one, because of course the effects of epinephrine, which is the same thing as adrenaline. So adrenaline is like what your body makes, but epinephrine is like what you get at the hospital, but they're the same thing. It's just where you're getting it from. So if we are giving somebody epinephrine and increasing their heart rate, you don't want to do that unless you really need to be increasing their heart rate and causing the effects of that. Also want to remind everybody that treating somebody with epinephrine is short-lived. It's enough time usually to get somebody to a hospital, but that reaction will return once that epinephrine runs out and is cleared out of the body, which is usually pretty quickly, within 30 minutes to an hour. And so if that allergen is still present, then they will revert back to anaphylaxis and then could be at risk of dying. So it's really important that if somebody needs to use an EpiPen, that you are taking them to the hospital, even if they feel better, because that won't last. So we can actually desensitize that IgE mast cell. We can actually prevent this from happening here by introducing things to trigger IgG production, so that if we have a little bit of IgE that are bound with mast cells, so this is just a little bit, but it's enough to cause your allergy symptoms. But if you're making, like we already said, if you're making IgG, you are making a ton of it, like way more than you will ever make of IgE, always, no matter what, like whatever the thing. So if you can stimulate IgG to see pet dander, that pet dander is never going to make it to cause this reaction. And so the idea is if I can stimulate and I can force your immune system to make IgG against pet dander, then maybe we will never have interaction with the IgE, and that can be long-lived. And so that is how the desensitization process actually works. All right, moving on to type 2. So this was number 1, was type 1. Type 2, these are all mediated by IgG or IgM. And this is essentially IgG or IgM binds to a different blood type, one that your body sees as foreign. If I have type A blood, my body sees type B as foreign. And so I have IgM or IgG against type B blood, and my antibody will bind to type B blood cells and cause complement to lyse it. Remember, one of the things the antibody does is activate complement. And so it activates the complement, causes that membrane attack complex to form, and lyses the foreign red blood cells. So that's how that works. This is going to rely on ABO blood groups. You either have A or B, or you might have A and B, or you might have none of them. So I always think as 0, O looks like a 0, that means you have none of them. And so 2A, if this is your type, what you see is foreign. If you're A, you see B is foreign. If you're B, you see A is foreign. If you're A or B, you don't see any of them as foreign. If you're O, you see both A and B as foreign. So if you are A, you can only get A, this is your blood type. So if you're A, you can get A and O. If you are B, you can get B and O. If you are AB, you can get A, B, obviously. You can get A, you can get B, and you can get O. That's all of them, right? Like, that's a pretty good deal. If you are O, what blood type can you get? O. Because you see A and B, you get AB, which is just both of them as foreign. So this is how this works. And so A and B are just different antigens that you can have on the surface of the cell, and O is just the lack of them. And so if you are an O blood type, you see A and B as foreign. Those markers don't look right to you. They look as something bad. The most common blood type in the United States is O positive. So where's the positive coming from? The positive comes from something that we call rhesus factor, also known as D factor. There's other names for it, but RH is really more commonly what's called the RH factor. And you either have it or you don't. If you have it, you have the antigen. If you don't have it, it's just like the blank slate, just like the O was. Okay, so if you're O negative, you can only get O negative. If you're O positive, you can get O positive and O negative. So having it is more common, and that's why O negative is our universal donor, because it lacks any of the antigens. Anybody can get O negative. Not anybody can get O positive. Only the positives can get O positive, right? So bringing that back. So basically what happens with RH factors in hemolytic disease of the newborn, which is the main thing that the type 2 thing brings home at the end of it, right? So yeah, blood transfusions, it's important. But with our RH factor stuff, RH, you're either you have it, you have the antigen, or you don't. No antigen. If you are a mother who is O negative and you are RH negative and your baby is O positive, it has the antigen, the mother and the baby. The mother who's carrying the positive will make anti-positive antibodies. It sees the positive as foreign. During the whole first time that she's pregnant, and the first time that her body starts seeing that blood, because your baby doesn't make blood cells and stuff like that until later in the pregnancy. That doesn't happen right away, right? We're just vague cells kind of multiplying for a while. And once we start having a blood type, the mother can see that as foreign and start making antibody to it. But the placental barrier prevents that from causing an illness. It's when the placental barrier becomes ruptured during pregnancy that the antibodies can start crossing over and attacking. So the whole time during the first pregnancy, the mom is just making these antibodies. This is her first sensitization. And so with the first baby that is born that is Rh positive, it's usually fine. It's usually fine. But the second time that the mother is pregnant with a baby that has the antigen, that Rh negative mother, her body sees that as foreign again. It doesn't need to go through the process of making the antibody. It can just start making it. And so once that baby is born, usually the second pregnancy or further for an Rh positive baby to an Rh negative mother, then that antibody will attack that baby's bloodstream and it causes massive hemolysis in the baby's body and commonly causes death of the newborn or stillbirth or death of the newborn after birth. This is still something that's very common today. And RhoGAM is an antibody that blocks that antibody from attacking the baby. So we can give RhoGAM injections to the mother before the birth and treat that mother so that the antibodies don't ever attack the baby during that birth process. So that's the whole point of RhoGAM, is to block this from attacking. All right, moving on. Type 3, this is immune complex related. Basically, like let's say you've had strep. Whatever. Here's the bacteria. Here's the strep bacteria, and it's got a capsule, whatever. It's nice, nasty little strep bacteria. And your body made antibodies to it. Cool. And you've got all these antibodies, and you've got little bits of strep and little bits of antibody kind of floating around in your bloodstream. And it creates what we call, and so we got more, well, little pieces of strep and little pieces of antibody kind of creating these complexes that get mixed out throughout your body. And normally your body will clear them out, but sometimes these get stuck in like your capillaries or in tight spaces in your body. And so these, when this forms, this is called an immune complex. It's usually because of IgM, IgG, or IgA, and they get stuck in these tight capillaries or in joints or in very, very narrow spaces like what we have with our filtration in our kidneys with our glomerulus. I can remember the words. Okay. I've never taken anatomy, so you guys have to, God, forgive me for this. So the glomerulus in the kidney, which is helping out with filtering out the blood and the toxins from the blood, very, very narrow capillaries involved in these structures, very delicate structures, and they don't withstand a lot of damage. The problem is that these complexes can get built up and caught inside of these narrow spaces. And then the body sees that as some sort of foreign reaction going on and starts activating complement or activating neutrophils to come and eat up this stuff and get rid of it or whatever it is. Your immune system gets activated because that stuff is stuck in there. And then we have inflammation as a result. So this is what causes acute post-streptococcal glomerulonephritis, which if you know these words and you're reading through them, it's a mouthful and I won't make you spell it out, but it is one of those things that you'll need to recognize for the test. So acute is because it's short-lived. Post-streptococcal, because it happens after a streptococcal infection, like what I just outlined here. Glomerulonephritis. Glomerulus is part of your kidney. Nephri means nephrus is Latin for your kidney. So your kidney tissue is becoming inflamed because these immune complexes got stuck in there. And the immune complexes exist because we thought we were binding with antigen from a previous strep infection, and it got stuck in our kidneys. So that's how that is caused. This is also the driving factor of what causes lupus and rheumatoid arthritis. Makes sense. Makes sense when you know anything about the presentation of these diseases. So in lupus, we have these immune complexes getting stuck in the capillaries of the face, causing that butterfly effect on the face with that rash. In rheumatoid arthritis, obviously the complexes are getting stuck in the joints. So that's how the autoimmunity relates there. SLE is lupus, rheumatoid arthritis. And then there's other ones that'll be involved in Graves' disease and type 1 diabetes. Basically, our immune system is getting triggered to act against our own self and our own body because of either type 1, 2, 3, or 4 reactions causing that stuff. So type 4 is our last one. It's our delayed and cell-mediated, and this is actually what drives our type 1 diabetes. So this is delayed because as we can say all we want with our memory, with our antibodies, you start making IgG right away. Yes, your memory B cells, once they get activated, can start making antibody right away. But your memory cytotoxic T cells, you've only got 1 or 2 memory cytotoxic T cells floating around. That's not enough to fight off a new infection, right? You don't need all of the signaling from all of your T helper cells and everything like that again, because we already know, we've seen the thing and we know how to react to it properly. But we still have to multiply back out our arsenal. So instead of taking a week or 2 to make our CD8 T cells, now we only take a couple days to reactivate our CD8 T cell pool. So your CD8 T cell that has seen, flu or something before, the reason that your vaccine might protect you from something like that would be because your antibody. But if you didn't have your antibodies 2 days into it, you would be protected by your CD8 T cells. So what happens is your CD8 T cells got activated in the past by something like poison ivy or something else. And those CD8 T cells remember seeing poison ivy. So that's why if you have poison ivy, you can go roll around in the poison ivy all day. 2 days later, you'll have a rash because 2 days later, your CD8 T cell is there saying like, this looks like you have something inside of you. Even though we know that actually practically that poison ivy oil is an outside thing, the way your body reacts to it, it sees it as an inside thing and presents it on MHC1 and causes cell death, self-cell killing. And that's actually why when you look at hives that you get from something that you're allergic to, like dermatitis wise, when you look at hives that you get from, whatever pet dander or whatever it is, versus the rash that you get from poison ivy, like the weeping and the oozing, and you have actual damage involved in that. They're quite different, the rash presentations there. So atopic dermatitis, you usually see rashes and things like that, whereas with this type of cytotoxic response, where we have reaction with contact dermatitis is the word that I couldn't think of. Contact dermatitis is usually this type 4 reaction. It's usually a couple days after you come into contact with it. I'm allergic to metals. And so cheap metals, if I wear them for a certain amount of time, usually not like the bracelet as much as it is like earrings, right? So if I wear cheap metals in my earrings, 2 days later, they get oozy and yucky and stuff, and it's itchy too. But it's usually a couple days after I've worn those earrings that I'll start having that reaction, because it is this inside stuff that's going on there. This is also the reaction that's involved in graft rejection whenever we reject an organ transplant. So it always requires an initial sensitization. You had to see it first and then become sensitive to it. And then every time that you see it, it's usually delayed by a couple of days. But this is also where we get host versus graft response, as well as graft versus host. And this is all driven by MHC1. I already wrote an H. Let's just commit MHC1. So again, this is when my body sees your MHC1 as foreign, and all of your body cells have MHC1. So anytime that you get somebody's new kidney put into a new body, my body sees that kidney's MHC1. And so if that MHC1 doesn't look like my MHC1, I'm going to see it as foreign, and I'm going to attack it. And we're going to start killing it with the cytotoxic T cell response. And so that is what drives organ rejection with the type 4 hypersensitivities. And so you usually don't see graft rejection for a couple days after you have put a kidney in. So those first couple of days after transplant are usually the most critical, and start seeing organ rejection. Usually people are put on medications to basically dampen the immune response. So you might not see it 2 days. You might see it a few days even later than that, because those people's immune systems have been treated to not have a response. And so this becomes very tricky, and that's why matching up people genetically is so important. Now, your liver can sustain a lot more damage, and it's not going to be as tricky. So usually liver transplants are a little bit more forgiving than kidney, lung, or heart. And bone marrow is completely different. So with a bone marrow transplant, you're usually removing somebody's lymphocytes from their body altogether. They don't have B or T cells that can attack anything. But the problem is that you're putting new B or T cells into somebody from somebody else. And so their B or T cells can attack you. And so when you get a organ or a bone marrow transplant, you're putting new lymphocytes into that person. They could see the person as foreign, and that would be graft versus host response. So more commonly with most tissue transplant, the host is attacking the graft, but with bone marrow, the graft can attack the host. All right, then there are different kinds of grafts that we learned about. The autograft is from yourself. Don't confuse auto and allo. If you're going to focus on anything, know the difference between an autograft and an allograft. All right, an autograft is from yourself. Allograft is from another person. Isograft is from an identical twin. And a xenograft is from another species. So this would be like heart valves from pigs or cows, for example. All right, we already talked a little bit about autoimmunity. Those autoimmune reactions can happen just as a result of these complexes building up, but it can also be because we didn't have proper clonal deletion during our upbringing, and that can be due to genetic differences. And also certain viral infections can actually trigger our body to accidentally target ourselves. They just give signals that are going to increase the likelihood of that happening. And then certain bacteria have markers on their surface that mimic us. That's called molecular mimicry. And that protects them from our immune system. But if our immune system decides to go and attack that thing, it can also end up attacking self as well. And then it also talks about changes in the gut biome. Our gut biome is absolutely the number one most important thing in training our immune system and how it's supposed to be reacting to things, when and where and how. So disruption of the gut biome can actually have a huge effect on your immune response, especially when you're younger, when a lot of that training is going on. All right, immune deficiencies, there's two, and this should be pretty quick. The two are primary. Primary immune deficiencies are from birth. They are genetic abnormalities. So genetic mutations, always from birth. And secondary is something that you acquire later in life. This can be always acquired somehow. This can be due to having HIV leading to AIDS, which is the actual immunodeficiency. Having HIV doesn't mean you have AIDS, right? But if you have AIDS, you had to have HIV to get AIDS, right? So, but having HIV, once your T cell count has lowered to a certain point, then we call that AIDS. Certain cancers can cause your immune system to be weakened as well. That can lead to immunodeficiencies, especially when we're talking about leukemias and lymphomas. Those are literally your immune cells that are involved in the cancers, and then now your immune cells aren't behaving the way they're supposed to, as well as radiation and chemotherapy. Your immune cells, because they're derived from your bone marrow, are actually pretty susceptible to being destroyed by chemotherapy or radiation. And so when you destroy those with the chemotherapy and radiation, that leads to the immunodeficiencies as a result of that. Where's my 18? Okay, do I want to do the immune thing? We'll do the immune response really quickly. We've kind of touched on these concepts, so it just shouldn't be too hard. So in your immune response, you first see, we'll do the virus again. We first see a virus outside before it will get inside, right? And so the virus gets into the body, it's still outside of cells. Okay. So this is our little line of the body. The virus is now inside. You've breathed it into your lungs, and it hasn't gotten into the lung tissue yet. Well, macrophages that we call alveolar macrophages in the lungs. So I'll abbreviate this as MPhage. Macrophages like this recognize a PAMP, like we wrote on the board. We're just going to review this. They see something about this. This looks like a bracelet. So we eat it up, right? Like we said in our example, we eat it up. And because it came from outside, we're going to express this on MHC2. It came from outside. All right. And this is that part of the virus. Well, as I was saying in my example, part of the bracelet. Does anybody recognize this? That's the question that it's asking when it does that. All right. So first things first, it's going to circulate around in the periphery. It might go to the lymph nodes, but your T cells are kind of circulating around in your periphery. So first things first, we get a T cell that comes in. All the T cells are coming and checking it out. Well, we finally find a T cell that sees this part of the virus, this antigen, with its T cell receptor. And it's just like an antibody. It's very specific to that antigen, all right? But always on a T cell, that T cell receptor is always attached to the surface. It's never going to get secreted. So this is a T cell. We know that for sure. And because this is being presented on MHC2, we know that this is a specific kind of T cell that has a specific marker on it called CD4. CD4 sees MHC2, always and only. MHC2 is only seen by CD4, and CD4 always sees MHC2. That's the interaction there. So because this is presented on MHC2, we know it's going to be interacting with CD4. So we know if it's a CD4 positive, it has to be a T helper. And right now it's naive. It's never been activated before. But because it's seeing this, we can now activate this. They're going to send signals back to each other, right? They're going to activate each other. How does that help the macrophage? It tells the macrophage, go forth and find more of this and activate other cells that are going to be involved in this. Keep up with the inflammatory response. We found a thing that doesn't belong here, and we need to keep fighting however we can. You keep going out and phagocytosing, and I'm going to start my end of my bargain, being the captain, right? And so when this happens, when the activation happens, we're going to first proliferate, or we're just going to be a whole bunch of THs. And then each of these that we've proliferated will differentiate. So this will become T memory cell. This will become a T regulator cell. and this will become TH2. And this will become, I'm writing in this order, so I'll have room on the other side here in a second. And then this will become TH1. TH1 and TH2. These guys here are what we call the effector cells. These are the ones that are actually going to do the job part. The regulator cell is just going to make sure things don't get out of hand. And the memory cell is just there to have memory. So if we ever see this again, we don't have to go through this whole proliferation and differentiation thing ever again. Because this part, proliferating and differentiating, this takes some time, man. This is where that week-long waiting is happening. Because it takes a long time for your body to make enough cells to have a proper response. All right, so great. So all that these guys do, these guys aren't going to help you with eating up a bad guy or killing a bad guy in any way. Not your T helper cells. They are just captains. They're just going to help make sure that we're activating the right responses at the right times in the right places. So I'm going to go to the next slide. Once you've activated your T helper cell and you've turned it into TH1 or TH2, these are still CD4 positive. So these still recognize MHC2. That doesn't change. But now let's say in the same body, you stop one of those viruses from going in. That macrophage ate it up. But that macrophage didn't eat up all of the viruses, right? So some of those viruses make it in to lung cells. And these lung cells get infected inside. All nucleated cells have the ability to signal on MHC1 for help. So what they'll do is say, oh my God, something's going wrong. Why am I making so much of this weird protein that I've never seen before? What is this? Does anybody recognize this? So it's the same question. Does anybody recognize this? Whatever this crap is that I'm making in here. Now, it can do this with any molecule inside of the cell. It can do this with, lysozyme or keratin or anything that could be inside of the cell, they can do this with. But normally when they do this with stuff that we have seen before from our own body, our cells will just be like, y'all, calm down. That is not a weird thing. And we've told our body that those are normal things. But now we've got a thing that isn't normal that we are presenting from inside. So if it came from inside, we know this is on MHC1. And it'll do this with any protein inside of the cell, any protein inside of the cell. But this time, this is something that our cells see as foreign. And as it turns out, somebody does recognize this. And it's another T cell receptor. This time, this one is going to be on our CD8 positive cytotoxic. And so MHC1 is seen by CD8, and CD8 always sees MHC1. So this is what happens, is this molecule on our CD8 T cell is CD8, interacts with MHC1. This all has to happen because the T cell receptor just sees the antigen, but if that antigen is presented on MHC2, that's not the right interaction. Cytotoxic T cell won't interact with it. It might still see the antigen, but because CD8 doesn't recognize MHC1 as there, we can't have the interaction. So we have to have all of these things happening, right? But this can't cause perforins and granzymes to break open this and cause it to lyse, which is also the same thing as break open and leak out its insides and die. We can't cause that to happen unless our TH1 activates it. It gives the right signals to this. We don't need to worry about the signals. I know the textbook talks about them, but I'm not going to ask you about the specific signals and what signals it sends and stuff like that. I feel like that would be like we're taking it too far at that point. So TH1 gives the signals to allow perforins and granzymes to be released at that point. But the TH1 had to get activated by the outside stuff, right? So you have to have both in order for this to happen. Now imagine on the flip side that you still have virus that's hanging out outside. Maybe this is because lung cells got infected. I'll even draw it. Lung infected, had viruses inside, and it's making copies of those viruses, and they're budding out and releasing new viruses that are found outside, and they're getting ready to infect the guy next door. Well, we know macrophages can eat these guys up. We've already kind of talked about that. But now let's imagine that these are floating around in your bloodstream, not just go into the next lung tissue, but some of this has made it into your bloodstream, or some of it's made into the tissue. And as the swelling and the inflammation is happening up, what happens to the fluid that's involved in the swelling and inflammation? It doesn't just go back into the bloodstream, right? It goes into the lymphatic system. So these guys have found their way into the lymphatic system. Which we learned about in your inflammatory response, all that fluid and all those neutrophils going out to eat up all these bad guys and whatever, that stuff is going to move back into the lymphatic system and filter through your lymph nodes before we rejoin the circulatory system. So these are now viruses that are outside into the lymphatic system, and you've got a B cell here. And it's naive. It's never been activated before. And it's got an antibody, what looks like an antibody on its surface, which we know as the B cell receptor, also known as IgD, type of immunoglobulin that acts as a B cell receptor. And it sees that virus. Oh, my husband's calling me at 12:30. It sees that virus and it says, I found this outside. Like your papers don't match. You don't belong here. And my IgD sees you specifically. You are, like we said earlier, Jason Voorhees. You're Jason Voorhees. I've been waiting for you my whole life. I'm a B cell that's been waiting for you. Maybe you saw your T cell over there, but I'm a B cell, and I've been waiting for you in this lymph node my whole life to see specifically you. So what it does is it takes this inside, because it found it outside. Again, this is MHC2. And it's going to take that part and say, I found this outside. Do you recognize this? At a military standpoint, if we're sticking with our military theme with the captains and stuff like that, on a military standpoint, this is a military guy saying, I found the target. Can you verify the target? And that's really what it's asking here. So what happens is, switch red, that same TH2 that we activated with the macrophage earlier can interact with this again. Again, this would be CD4. And because it's seen this before, it doesn't have to go through the double activating. It can just go ahead and give the activation signals to the naive B cell. If it hadn't seen it before and it was also naive, this would just be a longer process because we have to wait for it to become TH2 before we could actually truly activate everything else. But this one's seen it before. We saw it up above, right? We saw it in our other slide here. So now that we've activated our naive B cell, it's going to do the same thing that we did with our T cells. It's going to go through, proliferate into the different kinds of Bs. We'll have a B memory, a B regulator. It's going to proliferate to Bs. I should be more specific here. One. Proliferate into B, B, and B. Naive, naive, naive. And then differentiate. We'll be more specific here. We'll differentiate into B memory, B regulator, and plasma. Plasma cells are effectors, and then these guys will make first IgM, and then switch eventually to IgG and et cetera, like we discussed. Are we tracking? Does this kind of make sense? Okay. So this is the idea of how this will all work and all play out. And this is all happening kind of simultaneously, right? So as soon as we get that T cell activated, then all those T cells can go interact with the cytotoxic, and some of them are going and interacting with the B cells. B cells could have also activated different T cells that see a different part of the virus. We can be attacking several different parts of the virus with different kinds of T cells and B cells and all sorts of stuff. So this is all happening simultaneously, but the part that takes the longest is that proliferation and differentiation. You skip that with your secondary or amnesiatic response. You can just go straight to doing the effector job. So you get to skip all that with that. All right, so that's our review for the immune response. Moving on to chapter 18, and then we'll go through the questions really quickly. Yes. All right, phenotypic. We have basically 3 categories of tests. You are either phenotypic, immunologic, or genetic. If you are immunologic, we are dealing with antibodies and antigens. Genetic, we are dealing with literal nucleic acid sequence. We're going to go through some specific ones here in just a second, but I don't want to spend too much time on this one because we've gone through a lot of these tests before. But if you're dealing with anything about a characteristic, does it ferment? Like the test that we've been doing, right? Does it ferment lactose? Does it ferment lactose aggressively? Did it make carbon dioxide when it fermented, or didn't it? Those are questions that are dealing with phenotypic. What you're actually seeing that organism display as a trait, their characteristics, right? But if you're looking at the literal nucleic acid sequence, that's a genotypic or genetic test. If you're looking at antibody-antigen interactions, like what we do with ELISA, that's an immunologic test. Okay, so the whole point in all of this, so the really step-by-step diagnostic test-wise, you need to collect your specimen. Some of our specimens we can do direct testing on, and some of them we'll have to cultivate it first. And there's lots of different ways that you can collect different kinds of specimens, depending on what your patient's signs and symptoms may be. If you need to cultivate, we'll go through kind of the 5 I's in all of this. But the whole idea is to usually use an isolation media and or a selective or differential media, depending on what you're predicting. This also can include the use of blood cultures, which brings us back to that idea of septicemia versus bacteremia, where septicemia is actual flourishing of the microbes in the blood, right? We also might need to be considering the context of the organism in that disease and how that disease is presenting. And if somebody has a UTI, it might be organisms that are present normally in the normal biome in other areas of the body, for example. But if you have 1 or 2 E. coli, that might not be a concern. But if you have a ton of E. coli, especially in the urine, that's probably an infection. Whereas if you have even 1 Mycobacterium tuberculosis, that's probably an infection. You're not going to have that as part of the biome. You shouldn't be seeing any Mycobacterium tuberculosis. So the context of the infection versus the site of the infection versus the presentation of the patient all play into the idea of whether this is an infection that is worth pursuing medically. So our biochemical tests, like the ones we've been doing lately in lab, does it ferment? Does it make carbon dioxide? What biochemically is going on with our organism? These are all phenotypic. So in our big picture of phenotypic, a biochemical test is a type of phenotypic test. This is one that trips up a lot of people on that quiz, because it has some of those questions that are the same question, but different answers are available. And then how can those both be right? Well, that's because a biochemical test is a phenotypic test. And so if you're testing for, carbohydrate fermentation, is that a phenotypic test? Yes. But is it biochemical? Yeah, it's both. Just depends on what the question is asking and what your choices are. So be aware of that. Phage, like we've done antimicrobial sensitivity, before I skip over it, the Kirby-Bauer method, this is a phenotypic test. What were the characteristics of how your organism, behaved when we exposed it to this antibiotic? What were its zone of inhibition and things like that? That's a characteristic aspect. So that's a phenotypic test. We can also do phage typing. A bacteriophage only infects 1 species of bacteria. So it's a virus that only infects 1 species of bacteria. And so if I take your patient's stool sample and I culture it out and I expose it to a phage that only infects E. coli, and it infects your patient's bacteria, and your patient has an E. coli infection, However, if I treat it with a phage that infects salmonella and it doesn't infect it, then your patient doesn't have salmonella, but we can't say if they have E. coli or something like that, right? We don't know. We just know they don't have salmonella. But phage typing is using that specific virus that affects that specific organism. Moving on to the immunologic tests, many of these tests are serological in nature. And serological, again, is just another way of looking at immunologic. We're looking at an antibody-antigen interaction. The two major types of immunologic tests, which we learned about this in the context of ELISA, but this applies to literally all immunologic tests, whether they're indirect or direct. And it's just literally based on what question you're asking. Does the patient have the pathogen? If you're testing for the antigen itself, you're testing for the pathogen itself, then that's a direct test of that antigen. But if you're testing for antibodies to an antigen, using a known antigen to test for antibodies, right? We learned about this in lab, and that is indirect. So this term applies regardless of what immunologic test we're doing. We learned about ELISA, so I'm not going to talk about that again, but that's the idea. You can view it as agglutination or precipitation. Agglutination is whole cells. Precipitation is when we are causing antibody to cause molecules to stick together. So this would include like antibody interactions with toxins and things like that. Blood type testing, we're using whole cells, so that's actually true agglutination. All right, so immunochromatography is an immunologic test using antibody antigen. Chroma means color. Graphy means we're going to be moving across a paper, typically. Like you can rely on that. It's usually using a filter paper and rapid flow on a filter paper. These are our pregnancy tests and our flu and COVID home tests. If you're crossing paper and crossing a line and causing a color change, that's an immunochromatographic test. But it still uses that indirect or direct ideology. Antibody titer, we've talked about this previously as well. Antibody titer is a measure of how many of those antibodies are present, and that could be a sign of how recent or how distant an infection was. You can look for antibodies, but if they're in low numbers, it probably wasn't a recent infection. Specificity versus, you guys love this, right? Sensitivity, all right. You are sensitive, you pick up on things even in the lowest numbers. Specificity, you're picking on things that are only that antigen, not similar ones. So we know that you can take certain blood tests that test for certain illegal drugs that might test positive, even if though you're not taking that drug, you're taking a different drug. You can take cold medicine and pop up positive for methamphetamines, for example. And this is because they are chemically similar. That's because it's a low specificity. Specificity. It's not specific to methamphetamines. It's only as specific as drugs that look like methamphetamines. But the more specific you are, the more you're going to just see that specific thing. The more specific you are, the less likely you'll test positive when you're not positive. Same things with the drug test. The more specific the drug test, the less likely you'll test positive when you're not actually positive. Low chance of false positives. Sensitivity. You don't want to call somebody negative if they're actually positive. That person, when they do their home COVID test, the home COVID test is less sensitive than the PCR test, right? So the home COVID test might not pick it up. The home COVID test might not pick it up if you have one, a virus on there. But the PCR will, because the PCR is more sensitive. So the more sensitive you are, the less of that antigen that you need to be called positive. And then sensitivity will be a measure of that. And so basically like the likelihood, if you have 100 people that are infected, that will eventually show the disease. So 100 people that have the disease versus you only test 98 of them as positive, then the 98% is how sensitive it is. So that's a measure of sensitivity, right? All right. Next is in vitro testing. In vitro is in a test tube. In vivo is in like a living body. And there's in situ. In situ is where we would take like actual tissue from a body, like a biopsy, and then perform tests on that biopsy, but we haven't necessarily mistreated the cells outside of that. So it's kind of in between. So in vitro is complete test tube testing, in vivo is in the body, and in situ is somewhere in the middle, where we're still looking at the direct, you know, tissue sample or something like that. So some in vitro serology tests that we need to learn about for this. We've already learned about ELISA, so we don't need to learn about that. Enzyme-linked immunosorbent. Immunofluorescence also kind of speaks for itself. You're using antibodies that are basically labeled with a fluorescent molecule. And this antibody is blue because it sees COVID. And this antibody is green because it sees flu. And this antibody is yellow because it sees RSV. And then you can mix all of them together and then test your patient sample. And then whatever color your patient sample comes out as, you can say, this is this disease. So it's that kind of line of thinking with it. And then Western blot, we're basically running, you guys remember how we did gel electrophoresis, right? And we put the DNA samples in the wells, and then based on what lines you have, this might be the killer, or this might be the victim, or whatever, right? Well, proteins will separate out the same way. And so we can use electric current to separate out proteins, and then each band would represent a different protein, and then we can see if the antibody binds to that, and if the antibody does bind to that, it will cause a color change on a filter paper. That it's a lot of process that's involved in it, but you run the things out in the gel, move everything to a filter paper. If the antibody binds to it, then that person has that protein. Or you can also use the patient serum to do known things on the filter paper. And if the patient serum binds to it, then the patient has the antibody to the thing. But if it's separating it out on a gel and moving it to a filter paper, that's the Western blot. Okay. So Western blot, separate it out on a gel, move it to a filter paper, see if the antigen or antibody is there. That's Western blot. If you're using fluorescently labeled antibodies to see if the fluorescence sticks to something, that's immunofluorescence. And if we're doing that color change with the enzyme, that's ELISA. All right. Then we have the complement test. This is basically seeing if the patient has antibodies that can block complement from lysing red blood cells. If the patient has the antibodies, the red blood cells won't lyse. If the patient doesn't have the antibodies, then the red cells will lyse. And so it's just a matter of looking at lysis of red blood cells to see if they have antibodies that can block complement from lysing red blood cells. All right, so those are all immunologic types of tests. So we have kind of gone over the phenotypic tests. We've kind of gone over the immunologic. The main genetic tests, which we already introduced in Chapter 8, so I'm not going to spend much time on them, are going to be PCR, polymerase chain reaction, amplifying a known sequence of DNA, or not amplifying it if you're negative. Hybridization. This is using known DNA sequences and usually using in situ tissue samples. So you can take breast cancer, what you suspect might be breast cancer, take a biopsy of that and do an in situ hybridization with fluorescently labeled DNA sequence. So if the DNA sequence is fluorescently labeled, let's say it's green, and it sticks to your patient's breast cancer, then that patient, that biopsy turns green, and that patient has that nucleic acid sequence. So this is the same idea as that immunofluorescence, but not using antibody, using DNA sequence, and that DNA sequence is labeled. And so when that happens, that fluorescent in situ hybridization, it's called FISH. This is very commonly used to diagnose different kinds of cancers in situ. So we can take the biopsies, look if the nucleic acids bound. If it did, it was fluorescent, and that person has that genetic mutation. And microarrays is like using this, but like with a big old plate with like 364 different little DNA sequences on the plate, and then you mix the patient's serum or the patient cells onto that, and wherever the patient cells bind, that person has that genetic defect. We can do this for diagnosing diseases like infectious diseases as well. So we can test for a wide range of things all at once. Extremely expensive, but very effective if you need to test multiple things at once. All right, moving on to our other category. This is not phenotypic, genotypic, or immunologic. This includes mass spec. So this is actual chemical breakdown of what could be in your patient. This might be drugs, this might be actual pathogens, whatever, but we're literally shooting electrons at something and measuring how long it takes the broken off parts, the ionized particles, to reach a detector, which is called time of flight. And so we're literally shooting lasers at your patient's sample and then measuring how long it takes for that sample's parts to reach a detector. And then based on that, measures their mass. And we can know the exact molecular makeup of something using that. So that's how MALDI-TOF works. That's mass spectrometry. We can also use CT, MRI, PET scans, and X-rays to do imaging of your patient. That might not diagnose a cause, but might help you make procedures less invasive. Okay, let's do the questions real quick, shall we? All right. Yeah, you're good. The questions for chapter 16, obviously, knowing the differences between MHC1 and MHC2 is a major point for a lot of people. Have a good day. For a lot of people. So MHC1 presents the outside stuff, interacts with CD8. MHC2, and now I'm getting myself, I circled endogenous and I said the opposite thing. MHC1, the one looks like an eye, that's the inside stuff. So endogenous, inside. MHC1 presents the inside stuff to CD8. MHC2 presents the outside stuff to CD4, period. We'll write a little note out here. MHC1 presents inside antigen to to CD8. MHC2 presents out antigen to CD4. So which type of MHC molecule is primarily responsible for presenting antigens from intracellular pathogens inside to CD8 positive cytotoxic T cells? And that'll be MHC1. A patient with a genetic defect that affects the expression of MHC2 molecules would likely have difficulties with which of the following? Remember, MHC2 presents outside antigen to CD4. Will it have issues presenting inside antigen to CD8? No, that's what MHC1 does. Will it have issues activating helper T cells and orchestrating the immune response? Yes, because this MHC2 presents outside antigen to CD4. CD4 is found on T helper cells. And that T helper cells are captains that orchestrate the entire immune response. Will it have difficulties targeting cells infected with virus? That's an inside, so not that. Will it have difficulties eliminating cancerous cells? That's another inside, so it's going to be more B. Antibody production and class switching. So this is reminding us that B cells become plasma cells. They will clonally expand to the B cell that sees that specific antigen and then turn into plasma cells. We're going to start by making IgM and then class switch to make one of the other ones. Depending on what signals we're getting determines whether we make IgE, IgA, or IgE. IgG, did I say G? It doesn't really matter. So we're always going to start with IgM, which is the first antibody that we'll produce. This is going to be useful for making antibody complexes and activating complement, but it's not very useful for other actions of antibody that we learned about, like opsonization and things like that. Whereas IgG is our workhorse. It's found mostly in blood. It can be found in tissues as well, but it's the one that's going to be circulating throughout our body, and it's going to be the most abundant, and it can basically perform any antibody function. IgA is found in our mucosa. It usually exists as a dimer, and it looks sort of like this. And then there's another one. This is supposed to be a Y. All right. And they're kind of linked together in the middle, and then they combine with stuff out here. And so that's how IgA functions in the mucosa. It's what we call also a secretory antibody. Anywhere there's mucosa in your body, that's where IgA is going to be found. It won't really be found circulating in your blood and being found circulating throughout your body. It's mostly going to be found in the mucosa, secreted within the mucosal tissues. So this is what's going to be protecting the mucosa from pathogens. Whereas IgE is involved primarily is made for parasitic infections, but is also used in allergic reactions, which we learned about in chapter 17, which is going to be used by mast cells to granulate and cause inflammation. That's a useful response in parasitic infections, but it also leads to histamine release and signaling of activation by basophils, and then can lead to further activation of things like the eosinophils and stuff like that, which are involved in both allergic and parasitic infections. So our questions here, and I'll make my little notes. IgD was our receptor. It's found on the surface of B cells and acts as our B cell receptor. IgM is our first antibody that we first make. Then we will class switch to one of the other ones, IgG, IgA, or IgE. IgG, more commonly associated in the blood. It's our workhorse. It does most of the work. IgA is really focused only in the mucosa. It's the secretory antibody. And then IgE is more commonly associated with allergies or parasites. All right, our little notes there. All right, during antibody class switching, what change occurs in the antibody molecule? What's actually changing? We drew this out earlier in our notes. This is our variable region, and this is our constant. The variable region, also known as the FAB, the constant region, also known as the FC. The FAB region is what's going to be involved in antigen recognition, whereas the FC or constant region is what's going to be involved in the actual function and class of the antibody. All right. So if you are switching classes, what are we actually changing? We're switching classes. We don't need to change the FAB. What we need to change is our function in our class. That's in the FC, the constant region. So A, is it the variable region? No. B, is it the constant region? Yes. Let's read our other choices though. Will what be changing is the light chain? No, we don't need to change the light chain, which are these little pieces out here. We are changing the constant region. The light chain is part of the variable. Or will we be changing from a monomer to a pentamer? Actually, we'll be probably changing from the pentamer, the IgM, probably to the IgG, which is a monomer. So B is our answer for this question. A patient is diagnosed with an immunodeficiency that affects their ability to switch antibody classes from IgM to IgG. So they will be stuck with this IgM and they won't be able to go to the IgG. Which of the following would likely be true about this patient's immune response? The patient would have normal IgG levels, but low IgM? No, probably their IgG would be lower. The patient would have difficulty producing effective long-term immune responses against pathogens? Possibly. Let's look at our other choices. The patient would have high levels of IgE. That doesn't relate to the IgG. The patient would have an overproduction of IgA. No. So you have to get specific signals to turn to IgE or turn to IgA. If you can't make IgG, you don't just revert to the other two. So if you can't make IgG, you're just not going to have good IgG. And your IgG is what is key for your actual memory and protection stuff. So B is our answer here. Our next concept is understanding our difference between the T helper cell functions. So we have Th1, which moderate the inside response. They themselves are triggered by the outside, but their signals are going to help moderate the inside stuff, which needs more moderation because our inside response is basically driven by our cytotoxic T cells. And we absolutely cannot let our cytotoxic T cells start killing self cells indiscriminately. All right, our T helper cells, they are going to be more important in our outside. which is going to be focused more on our humoral immunity, which is antibody. Regulatory cells function to make sure that we are kept in control and don't cause autoimmunity or overstimulation. All right, so then we also have the memory cells that we would be making as well. So let's move on to our first question. Which helper T cell subset is primarily involved in promoting antibody production by B cells? Go back to our thing and I'll write a little note here. Th1 moderates our inside, which will be moderating our CD8 positive cytotoxic T cells. Our Th2 moderates the outside response. So we'll be controlling activation and antibody production of B cells. So this will be Th2. A patient with an autoimmune disease has elevated levels of pro-inflammatory cytokines and reduced function of T regulatory cells. So remember when we learned about our regulators? They're going to decrease inflammation, regulating inflammation at the level that it should be. We don't want it to be excessive. And it's also going to decrease the likelihood of autoimmunity. All right, so which of the following is most likely affected in this patient where we have pro-inflammatory cytokines, so increased production of inflammation and reduced regulation. So they're going to have an increased inflammation response and also have an increased incidence potentially of autoimmunity. So will this person have affected their ability to activate macrophages and cytotoxic T cells? It's not really what the T regulatories do. Will they have affected their ability to regulate the immune response to prevent autoimmune damage? This is exactly what we're talking about. Will they have affected the promotion of B cell antibody production and allergic responses? No, that's not really managed by our regulatory cells, T regulatory cells. It's going to be managed more on the B cell side. And then the suppression of inflammatory responses by eosinophils. Eosinophils are an innate. They are granulocytes on the innate side of things. They're not going to be affected by our T regulatory cells. So our B cell response, our choice B is our answer, regulating the immune response to prevent autoimmune damage, which is really what our T regulators are going to be doing. All right, moving on to chapter 17, most missed concepts, understanding type 1 hypersensitivities. We'll make our little notes up here. Type 1 is mediated by IgE with mast cell degranulation. These can be atopic, which are local, or anaphylactic. Which is systemic. And these are all basically based on what we consider to be traditional allergies. All right, so our first question, let's see if we can answer it with what we've got. Which of the following is the most immediate and effective treatment for anaphylaxis? So this is systemic. Most of the time when we're treating the systemic type 1 hypersensitivity, you want to treat it with a drug that has systemic effects, especially for opening up the airways immediately and leading to vasodilation, letting blood get back into the brain. And the best treatment for that, for systemic treatment, will be epinephrine, medically administered adrenaline. In the sensitization phase of a type 1 hypersensitivity reaction, what role do IgE antibodies play? So in sensitization, this is your first initial exposure to whatever you're allergic to. And so when you're going through this sensitization process, you're first seeing the antigen triggering your T helper cells, primarily your Th2, which will then activate your B cells, which will turn into plasma cells and will make the antibody. The antibody will first be IgM, and it'll class switch to IgE. This is your sensitization. right? So what role are the IgE antibodies going to then become? They're going to eventually bind to mast cells and just camp out in that area until they see the thing the next time. So the second time that you see this thing, that these mast cells will degranulate and cause histamine response and whatever. So what role are IgE antibodies playing? Are they neutralizing the allergen in the bloodstream? No. That is the hope of what IgG will do if you go through desensitization, though. In your actual sensitization process, are the IgE antibodies binding to the allergen-specific receptors on mast cells and basophils? Potentially this. Are they activating complement proteins to destroy the allergen? No. Are they enhancing phagocytosis of the allergen by macrophages? No. They are binding to receptors on mast cells or basophils. So when those mast cells or basophils, we're focusing on the mast cells, wear them as little receptors on their surfaces and they lie in wait for the allergen again, and then later release their stuff by degranulation. All right. Next is hemolytic disease of the newborn. This always happens in Rh negative mothers. We do not see this in Rh positive mothers. If an Rh positive mother has a baby that is Rh negative, that doesn't look weird to an Rh positive mother, but to an Rh negative mother, an Rh positive baby looks weird. And that's where this issue is going to be coming from. Okay, so we're going to write these notes out. Rh negative mother. First baby that is Rh positive will be the sensitization. We have now been alerted. Rh positive looks foreign. We've made antibodies to it. Anti-Rh antibodies. So the second baby that is Rh positive, we're just going to make our anti-Rh. We don't need to sensitize. And then this will lead to stillbirth or death. This is the hemolytic disease of the newborn. Babies can potentially be saved from this. Now, one of the questions that I get from a lot of my students will be like, well, if the mother is O and the baby is A, wouldn't the mother see the baby as foreign and attack the baby, and that could lead to hemolytic disease of the newborn? The answer is yes, but it's much less severe. The Rh is where we get the most severe reaction. So yes, if a mother is O and the baby is A or B, or if the mother is A and the baby is B, and it's the second time we're seeing B, or it's the second time we're seeing A, and we've never seen them before, right? It's got to be the second time because the first one is our sensitivity. So the second time you're having a baby that is foreign blood type of any kind, Rh or ABO, you can see hemolytic disease of the newborn, but the presentation is usually deadly with Rh. All right, so moving on. So which of the following is the primary method to prevent hemolytic disease of the newborn due to Rh incompatibility? What do we do to prevent this down here? We administer RhoGAM, which is an anti-Rho, Rh, also known as factor D, that blocks antibodies that are against the Rh. And we give this to Rh negative mothers. A newborn is diagnosed with severe jaundice, anemia, and enlarged liver shortly after birth. The mother is AB negative, and the baby's blood is A positive. So we've got the mother is AB. It doesn't look foreign to her, but that she's a negative. What does look foreign to her? The positive. This is the Rh factor. So what is the most likely cause of this? Is it because of ABO incompatibility? No, the A doesn't look weird to her. Is it because of Rh incompatibility? Yes. Is it because of viral hepatitis or maternal diabetes? No, doesn't relate to this. This is quite clearly due to that Rh incompatibility. So this is a scenario where the baby was diagnosed with the jaundice and severe anemia and enlarged liver as a result of the mother's antibodies attacking the body of the newborn. This is often deadly, but we can treat this after birth if we catch it soon enough and we act quickly enough. All right, chapter 18, our diagnostic tests. Remember our three major groups: phenotypic, these are characteristic based, or traits. Immunologic, this is antibody or antigen interaction. And genetic or genotypic testing. This is going to be literally based on nucleic acid sequence. All right, so our example questions, again, I'm going to write it down here. Phenotypic. It's going to be our characteristics or traits. Wow, characteristics or traits. Immunologic. This is going to be antibody or antigen. And then genotypic or genetic. Both words are correct. This is going to be nucleic acid sequence dependent. All right, so which type of diagnostic test would be most appropriate for identifying a bacterial pathogen based on its genetic material? So this is asking us, basically the question is, which of these tests is a genetic test? Gram staining, this is phenotypic. ELISA, this is immunologic. Polymerase chain reaction, this is going to be genotypic, genetic material related, right? And biochemical test strip, that's phenotypic. A patient undergoes a test to determine the presence of antibodies against HIV. So here, just going to point this out, antibodies against the presence of HIV. So what type of test is this going to be? Immunologic. Don't read too far into it. Okay, so we made it through our review for chapters 16, 17, and 18. Take some time to see if you can walk yourself through an immune response, because remember, this is coming back again for the final, which is just in a couple weeks. So you want to be sure that you've got this down pat so that whenever you're given scenarios for the final, not just for Unit 3 tomorrow, that we make sure that we're making sense of this stuff and taking time to understand it. We'll have our gameplay tonight at 8 p.m. And so we'll do that, just normal Kahoot gameplay playthrough. If you guys are there, bring your questions if you have any. I'll be watching as well to see how people are performing on the Kahoot and trying to point out questions that we're consistently getting wrong, kind of like I did for Unit 2. So I'll try to keep on top of that for you guys. And if you have any other questions outside of that, please email me. Take the time to make sense of this stuff. Walk yourself through it. And anywhere where you're getting stuck, just ask a question and other people will benefit from it too. So don't be afraid to ask. Other than that, I'm going to let you guys go. Have a good day. Email me if you have any questions.